Good afternoon, everyone, and thank you for joining us today for the award ceremony and the Distinguished Scientist Fellow Guest Lecture featuring Dr. Jerry Tuscan from Oak Ridge National Laboratory. I'm Dr. Linda Horton. I'm the Associate Deputy Director of Science Programs in the DOE's Office of Science, and I will be the moderator for today's event. I'm also pleased to welcome some of the DOE leadership, including Dr. Clyde Anderson, online, Dr. Jerry Richmond, and many others that I see here today from across the Office of Science. There are also DOE National Laboratory Directors and other National Lab staff online, including Dr. Steven Stryker from ORNL. We also believe that joining us are the nomination teams, the people that nominate folks for this award, the peer reviewers that assess the many high-quality scientists who were nominated, and of course our fellow, his family, and invited guests who have joined us. First, we have recorded remarks from Dr. Jerry Richmond, the Undersecretary for Science and Innovation for DOE.
Hello, everyone. Now, as Undersecretary for Science and Innovation in the Department of Energy, it is my pleasure to join you today and congratulate Dr. Gerald Tuscan of Oak Ridge National Laboratory for his selection as a 2024 DOE Office of Science Distinguished Scientist Fellow. Boy, do I wish I could be there in person today to give a more personal congratulations to you, Dr. Tuscan, and a big high five, as this is such a big deal. But being able to be part of this event that celebrates our best, even in just 2D, is a true honor and a personal joy for me. So as you will hear from others, Dr. Tuscan really represents the best of DOE, with over 33 years of scientific leadership and service at Oak Ridge National Lab and in the international biological and plant genetics community. Now, Dr. Tuscan and all of the Distinguished Science Fellows are why Secretary Granholm often calls DOE America's Solutions Department. They're strengthening American leadership and competitiveness in science and technology and advancing diverse fields in discovery science. Fundamental, groundbreaking science is baked into the DNA of DOE and to our success as a nation. So please join me in a short round of applause for Dr. Tuscan as I hand things over now to Dr. Linda Horton. Thank you, and congrats.
Next, we will hear from Dr. Steven Stryker, the Director of Oak Ridge National Laboratory. He's joining us today on Zoom. Steve?
Thank you, Dr. Horton, and I'm really sorry that I can't be with you there in person. This is really a fantastic event. I'm glad to say a few words. And thank you to the Office of Science for supporting our nation's top scientists in their groundbreaking research directions through what is really a very prestigious program. As everyone's aware, the Office of Science Distinguished Scientist Fellows are selected based on their outstanding scientific leadership, engagement with research communities, sustained scientific excellence and achievement, relevance to the programmatic goals of the DOE Office of Science, service to the research community, mentoring of early career scientists and/or engineers, and their commitment to diversity, equity, and inclusion. And as Jerry has already said, Jerry is really the epitome of all of these criteria, and Oak Ridge is really incredibly fortunate to call Jerry one of our own. As a corporate fellow, Jerry holds the highest scientific rank at ORNL, and he's spearheaded a crucial part of our portfolio through his leadership of the Center for Bioenergy Innovation. As you'll hear today, he's a pioneer in the field of plant genomics, and Jerry is always driving new possibilities in bioenergy, from resilient crops to sustainable fuels. And as Jerry would tell you, he doesn't do this alone, and I strongly commend him for building up the accomplishments of others as a committed and thoughtful leader. Likewise, he's an extremely well-regarded mentor who's training a new generation of scientists who will continue to solve the difficult and urgent challenges we face to create a more sustainable future. And as a matter of fact, this year, we're celebrating 20 years since the completed sequencing and assembly of the poplar genome, which was the first genome that was determined of a tree. And this was a significant milestone that Jerry coordinated for the DOE Joint Genome Institute. As then Secretary of Energy Spencer Abraham noted, by helping to lead this international collaboration to sequence the first tree genome, DOE once again is pioneering discovery class science that promises to yield important societal benefits. I look forward to hearing more about this during Jerry's lecture and hearing about the great impacts that have stemmed from this seminal achievement. One thing that Jerry's career embodies that I think we all can appreciate is the translational nature of science and the journey from exploring the fundamental questions to proposing world-changing applications. I'm confident Jerry's time as a Distinguished Scientist Fellow will lead to a continuation of his remarkable track record of discovery, communication about these great results to the broader public, a deepening of collaborations, and as Jerry pointed out, more solutions for the nation's problems. So thank you, Linda, and back to you.
Thank you, Stephen, for joining us today and offering such a nice summary of Jerry's many accomplishments. So moving on to the events of the day, it is important that we recognize the impact of research supported by the Office of Science. A great honor is celebrating the people whose careers have had such a big impact on the Office of Science and our programs. Today's guest speaker, Dr. Jerry Tuskin at Oak Ridge National Laboratory, exemplifies DOE's mission to advance our basic science understanding of biology, earth, and environmental systems to create a more sustainable and secure energy future. He is an Oak Ridge National Laboratory Corporate Fellow and the Director and Chief Executive Officer of the DOE Center for Bioenergy Innovation, an innovative 17-institution collaboration to create sustainable aviation fuel supply chains through the domestication of bioenergy-relevant plants and microbes. Thus, Dr. Tuskin, along with other accomplished senior scientists at the DOE National Lab, were selected as the 2024 DOE Office of Science Distinguished Scientist Fellows. The 2024 cohort includes Dr. Mary Bichet of Brookhaven National Laboratory, Dr. Lois Kerfman-McInnes of Argonne National Laboratory, and Dr. Christine Pearson of Lawrence Berkeley National Laboratory. Today's event is the second lecture in the four-part series for this year's fellows. Before the presentation of the award plaque and Dr. Tuskin's presentation, I'd like to say a few words about the fellows program and how the 2024 cohort of awardees was selected. The Office of Science has honored 17 fellows, including the 2024 cohort, since this program began in 2019. These awards honor the extraordinary scientific achievements and professional service records of senior scientists at the DOE National Laboratories. Each fellow receives $1 million in direct funding support to be used to support activities that develop, sustain, and promote excellence in Office of Science research through collaborations between institutions of higher education and the national laboratories. It's one of the unique aspects of this program. The nominations were solicited from all 17 of the DOE National Laboratories. Nominees were endorsed by the lab director, and then the selections was based on a rigorous internal and external review of the nominations. Each fellow represents excellence in their field and joins the growing community of fellows who represent the best of what DOE National Laboratories have to offer in science, mentorship, and service. Now it's my honor to tell you about today's honoree and speaker. Dr. Tuskin is honored for his influential contributions in leadership and bioenergy, including pioneering research in plant genomics, enabling a more sustainable future. His success in bringing together large teams of researchers from different disciplines and institutions, including collaborations with industry, has been instrumental in driving the scientific and technological process in this field. Since 2017, he has served as the Director and Chief Executive of the DOE Center for Bioenergy Innovation, CBI, at ORNL. You may not remember, I was on the red team for that initial proposal for that great center and saw it in its early stages of nucleation. Today, he leads more than 250 scientists from 17 university, industry, and academic partners to develop bioenergy supply for sustainable aviation fuels. CBI is one of the 4 Office of Science Bioenergy Research Centers. Each bioenergy research center seeks ways to improve the production of biofuels and bioproducts from biomass, and our key and moving forward the dynamic field of bioenergy research. CBI has produced over 1,600 scientific publications, generated 37 patents, and spun off 6 startup companies to commercialize the CBI discoveries. Very impactful for one of these large centers. Dr. Tuskin's role in managing and elevating bioenergy to the nation cannot be understated. And I have a few things on the impact that he's had. He established and led the international team of scientists in sequencing the first ever tree genome, that of the fast growing poplar tree, which is considered a primary energy crop for next generation biofuels. And I'm sure we're going to hear more about that. This achievement was featured on the cover of Science. His approach became a template for other international consortia focused on tree and non-tree species genetics and genomics. Dr. Tuskin also developed the first genome-wide association mapping effort and model population for woody plant species, enabling more sophisticated approaches to experimental manipulation of poplar and other tree species for the plant biology research community. His achievements in gene to phenotype discoveries have advanced several key aspects of bioenergy research, including development of feedstocks with improved growth and processing characteristics, identifying important plant microbe interactions that influence fertilizer usage and crop resilience, and exploring novel pathways for carbon sequestration in natural systems. He has made tremendous contributions to science, published more than 300 papers, resulting in nearly 40,000 citations. He holds 18 patents and has been recognized as a Battelle Distinguished Inventor. He's been recognized by his peers through fellowships, awards, honors, professional leadership positions, very many that we won't cite here. His leadership also extends to laboratory sequencing program at the Joint Genome Institute, the scientific user facility for that purpose at Berkeley Lab, where his contributions were critical to establishing JGI as a global leader in plant genomics. He has distinguished himself not only as a pioneering scientist in the field of bioenergy, but also as a dedicated advocate for diversity, inclusion, and education in science. His service to the community and future generations of scientists is truly inspiring. He has served on several governing committees focused on inclusion policies. He's mentored more than 30 postdocs and has established the CBI Early Career Fellowship Program that prepares young researchers to manage large interdisciplinary projects, one of those unsung skills that is so critical for success today. In his quest, gosh, there's a downside of using a computer for your notes for these things. I would need to move to paper copy as it has just left ahead for me. In his quest to broaden the impact of bioenergy research and make science accessible to everyone, Dr. Tuskin established CBI's educational outreach program, which aims to reach underrepresented and underserved communities through strategic partners, including the University of Maryland Eastern Shore and HBCU. Finally, and this is truly impressive, Dr. Tuskin and the CBI team developed a partnership with the Creative Discovery Museum in Tennessee to create a bioenergy learning curriculum tailored for middle and high school students, which I'll tell you from my experience with educational outreach, tough crowd. To date, this initiative has reached more than 300,000 students and families, many of them in rural communities. Dr. Tuskin clearly represents all the attributes of an Office of Science Distinguished Scientist Fellow, commitment to science, leadership, and service, and we are very pleased to honor him today. Next, we will present the plaque to Dr. Tuskin. I'd like to invite Dr. Todd Anderson from the Office of Biological and Environmental Research that has supported Jerry through his career to present this award to Dr. Tuskin. Jerry, please join Todd. It is our pleasure to present this award to Dr. Gerald Tuskin for his foundational scientific advancements in the development of hardy bioenergy feedstock crops. for excellence in leading large multi-institutional science teams towards a robust, sustainable bioeconomy, and for supporting the next generation of diverse scientists. Let's give Jerry a round of applause. And I haven't quite trained Katie to not have quite so many pictures.
It's just two.
Thank you.
Thank you.
And now the part we've been all waiting for. The best part of these events are not these wonderful remarks, which really commend people's careers, but actually hearing from the scientists and how their career in science has evolved. So Dr. Tuskin is going to give a lecture on the applications of populist biology, my journey to collaborative genomics research. Let's welcome Dr. Tuskin to the podium.
It is truly an honor and a privilege to be able to be here and present my career in research in popular genomics, which couldn't have happened without DOE support, specifically BER. So I want to thank right up front, thank DOE and BER for allowing me to develop such a exciting and thrilling career for me, and hopefully for you, as you see in the slide presentation. I'm going to give an overview of what is populous. I assume that some of you are not biologists in the audience here or virtually. So I'll talk a little bit about populous and why we chose it as a model species, and go into the natural history of poplar. How did it become the species we use today to help fuel the bioeconomy? And then a little bit about research developments over the last 10 or 15 years or so, and how poplar became a model species, and conclude the talk with a look to the future, the near future. What's going to happen in the next 5 or 10 years, or what I hope will happen in the next 5 or 10 years. So if we look at poplar, why did we choose populous as a model species? Well, first of all, poplar includes things like cottonwoods, aspens, and poplars that you may have heard of. It's native to North America, and it's one of the fastest growing temperate tree species in the world. So it circumnavigates the northern hemisphere. It occurs in Asia, in Europe, and in North America. It's undomesticated, which means that we have access to 100% of the genetic variation that ever existed in poplar. And we can use modern genomics approaches to mine that genetic diversity to create process advantaged or domesticated plant materials. You may not know, but plants typically have both male and female sexes in a single plant. But poplar's unique here. It's dioecious, meaning there are male plants and there are female plants. And this has impact on how you domesticate an organism, and I'll highlight what that means in a moment. It has a relatively small genome, and it is easily genetically transformed or engineered. So all of these features allow us to think about applying modern breeding, modern genetics, modern genomics approaches to accelerate the domestication of this organism. And then lastly, poplar is among the most diverse species on the planet. Because it is outcrossing, it's dioecious. You maintain heterozygosity, or big A, little a combinations of genes in the population. And that allows us then to create commercial hybrids that have unique performance capabilities. Poplar's earliest progenitor occurred about 470 million years ago, when aquatic plants emerged onto the terrestrial ecosystem. They found a rocky environment high in oxygen and lacking soil. But that organism carried with it the photosynthetic machinery. Photosynthesis is the process of capturing CO2 out of the atmosphere, combining it with water and creating glucose. And glucose becomes the building block of all things biological. So that early progenitor of poplar, which probably resembled slime mold on the surface of a rocky environment, carried with it the molecular machinery that we use today to fuel the bioeconomy. It had photosynthesis, which had evolved billions of years earlier through the combination of a bacteria with a future plant cell. And that process of photosynthesis then allowed autonomy. It allowed growth in the aquatic environment. By the time terrestrial plants began to emerge out of the aquatic environment, they carried with them the genes for pectin biosynthesis. Pectin is a carbohydrate or a polysaccharide that connects neighboring cells together. So the first land plants actually represented a combination of neighboring cells that formed an organism. It also carried with it the machinery for cellulose. Cellulose is just a linear chain of glucose molecules. It's the most abundant polymer on the planet. It exceeds all polymers made by humans, and it is the basis for our conversion into jet fuel. We use those glucose molecules, deconstruct that cellulose molecule to create a hydrocarbon for jet engines. It lacked lignin. Lignin is the glue that holds the cell wall together. To be able to survive the terrestrial environment, the plant cell had to be rigid. Because the aquatic environment supplied support, the plant cell wall provided the rigidity that allowed plant cells to exist in the atmosphere, but it lacked lignin. So you can think of lignin as the glue that holds the cell wall together. Stomata. Stomata are the pores or the openings in the leaf surface that allows gas exchange. So CO2 from the atmosphere enters the leaf, binds with water, as I mentioned earlier, and forms glucose. But those stomates also liberate water in what's called transpiration. And that becomes critical in the future as we're trying to develop these commodity crops for the bioeconomy. We're going to capture economically marginal agricultural land. We're not going to displace food production. And where are those lands? They're in the drought-stricken regions of the Great Plains. And so we need to control water liberation through the control of stomates, and you'll hear more about that in a minute. It lacked a vascular system for the transport of nutrients, and it lacked a root system. And the root system not only supplies structural stability to the plant, but it is the point of interactions with a lot of microbes. So plant microbe interaction occurs in the soil, where bacteria, archaea, and fungi interact intimately with the plant roots to provide benefits, such as nutrient uptake or drought tolerance. Lignin did ultimately evolve in about 360 million years ago. And so lignin's been around a long time. And I said it was the glue that holds the cell together, but lignin allowed the plants to obtain tree form. It allowed arboreal habits, upright growth. And there was no organism on the planet that could deconstruct lignin. And so lignin was synthesized, the plant lived, the plant died, and the carbohydrates, cellulose and hemicellulose and pectins, were degraded by fungi and bacteria. But the lignin accumulated on the surface of the planet. And for 60 million years, this lignin piled up on the surface of the planet. And that's what's known as the Carboniferous period. And ultimately, it formed the layers of carbon that we burn today for energy. So that CO2 that's in coal came in through the process of photosynthesis, was fixed, formed coal, and we now use it for energy. Can we do something different? I think we can. Ultimately, a fungus evolved that could eat lignin. And so those lignolytic enzymes are the exact same enzymes we use today to deconstruct biomass and make jet fuel. So without that biological foundation of lignin formation and a fungus eating lignin, we would have a very difficult time today utilizing lignin. Lignin became so abundant on the surface of the planet that from outer space, parts of the Earth looked black. There was so much lignin accumulating on the surface. Poplar began with a bang. When the asteroid that impacted the Earth about 70 million years ago, known as the KT boundary, wiped out dinosaurs, it also wiped out early plant life. The early gymnosperms and ferns were destroyed in the impact. And this created an open environment, a niche, where poplar could evolve. The first record of populus was about 44 million years ago. And today, we have about 30 species of poplar. And I work primarily with Populus trichocarpa, which is native to North America and occurs along the Pacific coast from Alaska all the way down to Baja, Mexico. So when did poplar enter the human historical record? It is first recorded in early Rome. The name populus translates literally as the people's tree. And Romans used poplar to create a habitable urban environment. Poplar has the ability to take a dormant, unrooted stick, drive it in the ground, and it'll automatically root and grow and form a canopy. And so the Romans distributed sticks to urban settings to create an urban forest. So I would advocate that without populus, we wouldn't have Western civilization. So poplar played a critical role here. Next, during the Napoleonic, Napoleon was having trouble marching across Europe in the winter because he could not identify where the road systems were. So he challenged his engineers to outline roads so that he could invade Prussia. We all know how that turned out. But his engineers used the same technique that the Romans did. They took unrooted sticks and drove them in the ground and lined roads. And this is a sketch, a Van Gogh sketch of those poplar-lined boulevards that still exist today. Then Lewis and Clark and the expedition that discovered the Pacific Northwest for European settlers. Lewis and Clark used cottonwood that occurred naturally along their route in the Missouri, Snake, and Columbia rivers to carve out canoes. And it allowed them to travel faster using these waterways. So again, without populus, we may not be a nation from sea to shining sea. Poplar helped facilitate our exploration of North America. Then the Battle of the Alamo, in Spanish, Alamo means cottonwood. And so the mission was known as the Alamo because there was a giant cottonwood there. And so without poplar, we may be saying, remember the post oak, which just does not sound quite as attractive as remember the Alamo. My path to poplar and genetics. So my dad and my brothers and I would camp along the Verde River in central Arizona, and we would sleep under a giant Fremont cottonwood. That feral looking 14-year-old on the right is me. And somehow I learned to love cottonwood as a result of the experience my father provided me and my brothers. My undergraduate degree then was in Arizona. And as a sophomore, I took a class in genetics. And that changed my entire course forward. Dr. Smith was so enthusiastic about the potential of genetics that without taking that class, I may not be here today. So I owe a lot to him. Finished my degree in forestry and went on to get my master's in forest genetics at Mississippi State. And my master's program was funded by DOE. It was funded at the time by EERE, looking at genetic diversity and biomass production in sycamore. My PhD was at Texas A&M, where I continued, got my PhD in genetics and applied that to intergenotypic competition and its impact on yield. So if you grow a monoculture of corn and alternate every other genotype with an alternate background, how does that impact yield? Was the premise of the dissertation. And then my first job was in North Dakota State, where I was hired to work on poplar as a shelter belt system to prevent wind erosion. And so somehow, not beknownst to me, poplar was pulling me into its genetics. I was hired then at Oak Ridge National Lab by Janet Krishman and Lynn Wright, and they ran the Biofuels Feedstock Development Program, which was a BETO-funded program. And so my job was to run the woody crops program, evaluating production and productivity. We ultimately ended up sorting through the various yield trials, and you can see the distribution of the yield trials at that time were fairly widespread. And poplar rose to the top. And poplar was the fastest growing, most widely adapted, most amenable to biochemical conversion into biofuels. We didn't have access to the genome at the time, but we did know that DNA and genetics impacted productivity. And this is just an early presentation of the effect of genetics on stomatal size, those pores that allow CO2 and water to move in and out of a plant. Because we were developing genetic markers, the next project I worked on was a post-Yellowstone fire, where for the first time in the park's history, they witnessed regeneration of aspen. So for 100 years, they had never seen aspen seedlings. After the 1988 fires, aspen repopulated the park. And they hired Monica Turner, who led the project, and me, Monica as an ecologist, me as a geneticist, to come in and characterize where did that material come from? What was the genetic origins of it? And so we applied the DNA markers that were developed, and we determined that most of the seedlings in the park came from fertilization beds outside the park. And so the fire actually was a healthy thing. It improved the genetic diversity within the aspen population, which is a keystone species for other organisms. We then, my next project was working on the Chernobyl exclusion zone. So there were 3 eruptions in Chernobyl. The first 2 went to the west, and you can see those depicted here in the heat map. And the 3rd explosion went to the northwest, which triggered the alarms in Sweden. But as a result of those 3 explosions, cesium and strontium were distributed across the environment, which led to the exclusion zone that you see represented here. And it's hard to see in this graph, but the Napier River runs right through here, which is the water source for Kyiv and Odessa. And so DOE offered technologies to phytoremediate cesium and strontium. It was known that poplar has an affinity for cesium and strontium. And so we established willow and poplar double row crops to interrupt the cesium flow in the groundwater into the Napier River. And that's me and Tom Early standing outside the Chernobyl office sites. Tim Czaplinski and I became colleagues early on in our career. Tim is a plant biochemist, and he works on secondary metabolites. And what a secondary metabolite is a molecule, a biochemical compound that occurs within leaves, roots, and stems that affect plant microbe interactions, UV resistance to high intensity light, and drought tolerance. And we began looking at the accumulation of osmoticum or metabolites within the leaves as a mechanism for drought tolerance. And we published several papers on that. Tongming Yin then joined the lab, and we began developing microsatellite DNA markers. These are markers that are distributed across the genome. The pie chart here represents the 19 chromosomes of poplar, and you can see maybe, hopefully, hundreds of microsatellite or DNA markers were distributed across the genome. And once we built this library, we applied it to understanding gender in poplar. What determines maleness? What determines femaleness? And it turns out that the loci or gene controlling gender is a single gene located on the terminal end of chromosome 19. Without those markers, we would not have been able to identify that. I was then brought into the Joint Genome Institute and worked with 2 very hardworking, bright scientists, Jeremy Schmutz on genome assembly and Dan Roxar on genome annotation. And we sequenced and assembled the first tree genome. This was right after the human genome. DOE had the capacity to do sequencing, and they were looking for the next application. We stepped in front of a panel in a room like this and pitched the idea of the poplar genome. Ari Petrinos was leading the effort at the time, and Craig Bettner was there and slammed his hand on the table and said, "Ari, that's the one you should do." So Ultimately, poplar became the next sequenced genome. The poplar genome was the first plant genome to be assembled by shotgun sequencing. What that means prior to this was you'd have to clone DNA into a bacterial library. And that cloning process, then you would multiply the bacteria and sequence the DNA that was cloned. But the shotgun approach, you just take leaf tissue, macerate it, isolate the DNA, and then sequence small fragments of it. So this was the first combination of computational approaches and biochemistry to create an assembled genome. Today, all genomes, all plant genomes use shotgun sequencing. We were the first to assemble a genome at chromosome scale using genetic mapping. And we were the first report of a plant microbiome based on sequence information. This information is in the science paper, but you'd have to turn to the appendices. Now, we sequenced leaf and root tissue, and I told you macerate it and sequence DNA. Well, as a result of that, we also sequenced the bacteria, archaea, and fungi found in the leaves and found in the roots. But the reviewers did not believe it. The reviewers said, plants don't have that many microbes in their tissues. It's just not possible. Fast forward 20 years later, and every one of those genera have been proven to be endophytic inside the leaves of poplar. And so we really, I think, had uncovered a whole new approach to understanding plant microbe interactions. We were also able to prove that poplar evolved as a result of the molecular clock, prove that poplar evolved right at the KT boundary. Now, molecular clocks, what does that mean and why is it important? Well, if you're a taxonomist and you want to study the relationship between two neighboring plants, two species of poplar, you look at their DNA, and at the time it was assumed that the molecular clock ticked at a constant rate for humans, for animals, for plants. But we were able to clearly demonstrate that the molecular clock in perennial organisms ticks at a slower rate. And so you can't apply a uniform ticking rate on the clock to determine how long ago two species shared a common ancestor. Jeremy and Dan and I went on to sequence several other plant genomes. Foxtail millet as a switchgrass comparator, Brachypodium as a switchgrass outgroup. Switchgrass is a bioenergy model species. We sequenced Kalanchoe with the addition of Shohan Yang's expertise in synthetic biology because of CAM photosynthesis. It's a project that was funded by BER, looking at how do those stomates open and close at day and nighttime periods. We sequenced eucalyptus. Eucalyptus happens to have the largest array of terpene biosynthesis enzymes. Terpenes, you get eucalyptus oil or tree tea oil, you use it for medicinal or cosmetic purposes, but it also has value as a feedstock for commodity chemicals and fuels. So we were interested in terpene biosynthesis. I'll talk more about that in a minute. And then finally, Dan and Jeremy and I were able to prove that gender is a single locus using willow as an outgroup. In addition, we sequenced a whole series of associated bacteria and fungi. There are pathogens, there are beneficial bacteria, and these are listed here. Steve DeFazio and I set up the poplar GWAS study, sampled individual genotypes from across the Pacific Northwest, clonally propagated them into a common garden using the same approach that the Romans used. We took bear sticks, stuck them in the ground, and you could clonally replicate all 1,500 genotypes. JGI, Dan, Jeremy, and I resequenced all of those, and we created a large DNA marker set. This is the most informative, high-resolution genetic library of any plant species publicly available in the world today, which means we can link a single mutation at the DNA level to changes in protein structure at the phenotypic level. We applied those techniques and began to identify genes related to bioenergy. Wellington Lucharo and Jay Chen joined the team, and we discovered a gene that controls lignin deposition. It's called EPSP synthase. And it occurs about 35 biochemical steps away from lignin's precursor. It turns out that that's a regulatory protein. It determines the production of lignin downstream and removes carbon from the flavonoid pathway into the lignin biosynthetic pathway. It does this because it has co-opted a bacterial domain, the HTH domain in red here, which is a nuclear trafficking and DNA binding domain. So we think of ourselves, we think of other organisms as these discrete, isolated sets of genes that determine performance. But here we demonstrate that it is actually the intimate relationship and the exchange of DNA between organisms that leads to unique functions. And that message will come up again. We also discovered a gene that controls microbial colonization. Mycorrhizal fungi form intimate relationships biochemically with their plant host. You may or may not be able to see here, but this is Arabidopsis, and Lacaria produces hyphae. And in the absence of the LRK, the hyphae grow along the root system and don't even acknowledge that the root system exists. But in the presence of this poplar gene, now introduced into Arabidopsis, Lacaria colonizes the root system and changes its behavior. Prior to this paper being published, the common knowledge was that it takes hundreds of genes to allow colonization. Here we isolated a gene from poplar, introduced it into Arabidopsis, which has no known mycorrhizal associates, and induced functional colonization. It changed the way we see the intimate relationship between plants and microbes. We also began, Wellington and Jay and I began looking at bark roughness. And you would say, well, that's esoteric and interesting, but why bark roughness? Bark contains antibacterial and antifungal compounds that protect a tree as it's growing. Some genotypes have smooth bark, some have very rough bark. So we mapped that so we could identify or minimize the amount of bark on a harvested stem. It turns out that that is a protein, a single protein that acts like Velcro. It has a hairpin structure, and the hairpin structure holds on to its neighbor. And as a result of that, the bark is retained over time. It creates that rough bark appearance. If there's a mutation, a single amino acid mutation where the protein is flat, bark is sloughed off and you get the smooth bark. It turns out that that gene is the same gene, the exact same gene that causes keratoderma in humans. But in humans, the wild type is the flat protein, and the mutated one is the hairpin by a single amino acid substitution. We looked at callus induction. Callus is the unorganized rapid division of cells during wounding. So if a plant is wounded, it forms callus, but that callus is the vehicle for genetic engineering. It allows agrobacterium to insert a message, a piece of DNA, into the cells. The genes that control callus are the same genes that are associated with 5 types of cancer. It's the uncontrolled proliferation of cells. I fast forward now to the Center for Bioenergy Innovation. With our mission is to domesticate plants and microbes for the production of bioenergy related feedstocks for aviation fuel, chemicals, and materials. I highlight Brian Davison and Renee Speck, who are the Chief Science Officer and Chief Operating Officer. It's a large center, we've heard about that. And I just want to highlight a development that is hot off the press, still hasn't been published, where we discovered a gene that co-opts a piece of the photosynthetic equipment, combines that naturally in the population with DNA from a bacteria and DNA from an ant that then functions as a regulator of photosynthesis. And overexpressing this gene, this bacteria ant photosynthetic chimeric gene, you increase productivity by 200% over 90 days. And we've cloned that, we've put it in Arabidopsis, we've put it in rice, in tobacco, and you see similar effects. And we have a patent application for them. phenotyping becomes the next limiting thing, and we've developed a high throughput phenotyping system. I just draw your attention to the picture at the bottom. This is a single plant, but in hyperspectral space. And so to us, it would look like a rosette of green leaves. But under hyperspectral light, you can see that there are sectors of the plant that are behaving differently. We're using this to understand biochemical responses to drought, nitrogen use efficiency, pathogenic attack, favorable microbial colonization. Ultimately, we've successfully developed feedstocks from lignin, from cellulose, from hemicellulose that have passed the ASTM certification for jet fuel. And so we know technically we can now take that biomass using the genes that evolved 400 million years ago, deconstruct that into a carbohydrate that is catalytically upgraded into a hydrocarbon that functions and meets all the 9 parameters for jet engine production. And I just highlight the contributions of Greg, Yuri, and Deb here. With the new project, I want to thank DOE for giving me the opportunity to initiate new work in eucalyptus. I mentioned earlier that eucalyptus contains the largest number of terpene biosynthetic genes. We're going to utilize that biology to create chemical feedstocks. And this couldn't have happened without investment from DOE, for which I'm very grateful. I think there are other areas that are on the verge of really changing the way we see biology. We're beginning to look at single cell genomics, single cell metabolomics, single cell proteomics, because a leaf is a combination of multiple cells and tissues, and those cells communicate to each other. So we now have the ability computationally, technically, to look at the expression of genes in a single cell, whether it be from a stem or a root or a leaf. And this is going to change the way we see biology in the next 5 years. We need to do a little bit better job in species-specific functional annotation. Our annotation meaning the name or function of a gene, is determined by what Arabidopsis does. And that is determined by what yeast does. And so we project all this information into poplar, into switchgrass, into miscanthus, and we're wrong a lot of the time. And so we need to do a little bit better job in sampling the tree of life and looking at functional gene annotation. And then finally, there are hidden genomic structures that we don't know about. And we're going to uncover those in the next 5 years using large language models, applying AI and machine learning to tease apart these structures that are affecting the way genes are turned on or turned off. With that, and in closing, the award is given to one person, but really it represents a combination of very bright and hardworking people. I became a better biochemist when I worked with Deb Monin and Tim Chaplinsky. I became a better project manager when I worked with Lynn Wright, Janet Cushman, Brian Davison, Renee Speck. I became a better ecologist working with my colleagues. And that opportunity was a result of DOE's investment. In closing, the 21st century has been called the century of biology by lots of people. We are currently poised, thanks to DOE's investment, to achieve bold discoveries and disruptive technologies that are going to create domestic jobs, reduce our dependence on foreign oil, and help evolve the emerging bioeconomy. With that, I want to thank ORNL for giving me the opportunity so many years ago to become a scientist that I am today. The Joint Genome Institute for hiring me, the Department of Energy. I am the boy that BER built. And so thank you very much, and certainly for the Center for Bioenergy Innovation. Thank you.
Thank you, Jerry. And now we're going to confuse, I'm sorry, I'm going to stand here. Are there any questions from the audience?
I was curious in the story about the paper and the microbiome. You said that the reviewers didn't believe you, so what happened? You had to just publish a table and not say where they came from, or is it they were not
We
mentioned?
described, at that time.
You might
the way
repeat-
science
Repeat the question because I don't think the audience.
So the question was about the cover issue of Poplar, the first tree genome in Science, and the microbiome that we discovered using shotgun sequencing. The reviewers didn't believe, they thought it was contamination, and they didn't believe they were endophytic, that they occurred inside the cells, leaf tissue itself. And so they made us move that table out of the main paper and into the appendix. It's Appendix 27, not that I follow that. Appendix 27, and then you could have a narrative. There was actually, at that time, you could write a narrative for each of the tables. And so we described it in the appendices, but it would, despite our argument that it belonged in the main paper, it got moved to the appendix. It's a good lesson for early career scientists. You know, you may make discoveries that you know are bona fide and real, but you have to convince a panel of reviewers or peer reviewers that it's real. And this ended up, we didn't convince them, but in the long run, we did demonstrate that all those fungi, bacteria, and archaea are found inside the tissues of Poplar.
The other lesson is you documented it in the paper, even if it
Yes.
was an appendix,
Yes.
so it's archive. Sharon, do you know?
He actually took my question.
But anyways, I have first.
Other questions? Also, if you're online, you can use the Q&A feature to submit questions, and then Todd will pass them along as appropriate. So, yes, sir.
What extent of some of the species you mentioned have problematic other aspects, like eucalyptus is a water hog. So
Yeah.
to what extent do you the terpenes are great, but to what extent do you work on the other aspects of these species
Yeah.
that have trouble?
So can I layer on other aspects of problems with eucalyptus? Because I've thought about these. So it is known as an invasive species. It's known as a water hog. It's known as being not winter hardy. So why in the US would we plant eucalyptus? What I didn't mention on that slide was that we're going to test this and deploy it under what's called a coppice system, where you harvest every year. So it is only grown as a shrub. It's not grown as a tree. It won't flower, so there's no invasiveness. Its root system is insulated by the soil, so there's no winter hardiness. And as an annual harvest, it does not consume as much water as a giant tree. We think that we can deploy this. We've done the techno-economic landscape impact assessments all the way up to central Alabama, Mississippi, and down through Florida. We think that it will produce up to $500,000 in revenue for this region if we can get this system successfully working in the Southeast.
I have another
Yeah.
question. The study that you had done with the smoothness and roughness of bark, and then its application towards perhaps a human treatment. Can you talk a little bit about what the process of going from that kind of a result to an actual human-based?
So the project was funded through BER, and our goal was to characterize the molecular mechanisms of this phenomenon of holding on to neighboring dead cells, as bark is dead, actually. And so once we discovered that mechanism through homology, we began to find that it is the exact same gene that's known in humans. Because our mission space didn't include human work, that's where our involvement stopped. But we've published a paper. We received tons of inquiries from NIH and other scientists who have begun to work on this. I could offer one more one more outcome that I didn't publish. Poplar leaves are symmetrical, they're heart-shaped. And we noticed that some individual genotypes in the population had asymmetry. And that asymmetry led to lower photosynthetic capacity because the amount of leaf area was reduced. We mapped that gene, and it turns out that that gene is related to the exact same gene that causes DeGeorge's syndrome in humans, asymmetrical division or cleft palate. They'd known that gene in humans for years, and they'd used mouse models to try to understand what was the actual cause of this. Because in poplar, we have such high resolution, we could pinpoint the amino acid substitution that led to the change in the symmetry of a leaf, and that same mutation leads to the DeGeorge's syndrome in humans. And so that work stopped with photosynthesis, because that's what our mission space is. But that work has continued on in other areas.
From the online questions, Scott?
We do have a couple. Here's one here. You mentioned that you worked on projects funded by both BER and BETO.
Yes.
How do you approach the research differently from the different sponsors?
Yeah. Early in my career, I was funded solely by BETO, and we looked at applications of plant materials distributed across the US. So we weren't actively involved in the basic science of what caused those plants to be superior. We knew that poplar grew well, we knew that willow grew well, silver maple, black locust, etc. And through just establishing these trials across the US, we identified poplar as being superior. That was a role that BETO played. But when it came to the basic science, what makes yield yield? What makes disease resistance resistant? That's where BER stepped in and began funding basic science understanding of the mechanisms that led to these improved growth traits.
We do have another one here, a very good one. What do you think is currently the most limiting or challenging aspect of scaling up the biomanufacturing process?
That's a very good question, and I've thought about it a lot. There has to be, if we're going to impact the economy at a national level, there has to be landowner adoption of these new production systems that are, if it's switchgrass or miscanthus or poplar or willow, that are different than growing corn or soybeans. So landowner adoption is a barrier that's in front of us. And then on the conversion side, we estimate that to displace 10 billion gallons of annual jet fuel consumption, we need between 10 and 15 biorefineries, each at hundreds of millions of dollars. So there's an obstacle in front of us on investments in building these facilities. We play a role right in the middle of both of those. We're trying to develop processed advantaged feedstocks that you could deconstruct economically. But then a company would have to adopt that and do pilot scale research to get it to the level where we could commercialize it. And likewise, we could develop these production systems where we know we have nitrogen use efficient materials, drought tolerant materials, but they have to be adopted by landowners. That's outside our mission space right now.
I have one very practical question. How long will it be before I can fly a commercial flight fueled by plant-derived jet fuel?
Well, that's a great question. If that person has flown out of LAX, you've flown on sustainable aviation fuel. It comes from fryer oil. So about 3% of the fuel at LAX comes from fryer oil. That's not, you can't do that everywhere. LA must eat a lot of French fries. I don't know. But that's the first round. We're going to look at lipids and fats to create aviation fuel. The next round will be converting ethanol into hydrocarbons. And the third phase of this will be in lignocellulosics, and that's poplar and switchgrass and other biomass. So we're flying on aviation, plant-based aviation fuel now, but it should only increase and expand in the future.
All right, well, I have some of these other questions that
Okay.
are fun ones.
Okay.
But, so.
These have all been fun.
Oh, yes, they have been, but these are, And what comes from, and I'm just paraphrasing, I know that we shared these with you before. But you talked about the outreach and bringing in all these different community groups and,
Yes.
and working with people that are not in the normal fields that. How has that influenced your science?
Yeah, let me first of all give credit to Brian Davison, who helped form the relationship with the Creative Discovery Museum. And they have deployed these education sets out to middle schoolers and reached hundreds of thousands of folks over the last several years. What that allowed us to do was engage teachers. We would present a lesson kit, a lesson plan, and then they would say, you know, you're talking up here, but you need to be down here. And so it has helped us translate what we do technically, scientifically, I hope, into something that's easily understood by a sixth grader in Wyoming who happens to dial into the lesson plan. And we've had really good feedback on that and a lot of good interactions with middle school teachers that have helped us craft those messages.
Have you found that that those interactions have influenced the direction of your research at all? Or I get the part about engagement of people and
So as part of the outreach, we have a program called Talk to a Scientist, where anybody during an online lesson plan can ask questions. And those are always very interesting. You get questions from left field, from outside the ballpark, and that has helped us evaluate how we view the work we're doing and how we're hopefully moving forward in the work we're doing.
interesting. So you've spent a lot of your career at Oak Ridge National Laboratory.
Yes.
What would you say about that? How did that influence your science?
Yeah. You know, I came out of, my first job was at North Dakota State University. The opening came up at ORNL. I applied and was lucky enough to get the job. And I thought to myself, I'm going to come to the lab. I'm going to work for 5 years.
I give you 3 and 5 years.
And get some experience. I'm going to go back to academia. And very quickly, it became apparent that I would become a better scientist in the environment in which the national labs create. And I very early got to work with atmospheric chemists when the UVB radiation work was going on. There was a question about disruption of pollen germination. Pollen blows in the air, it lands on our cars, we hate it, we wash it off. But that pollen is exposed directly to UV radiation. And so talking to the atmospheric physicists and the atmospheric chemists, we designed an experiment to assay, will pollen germination be affected? Will thymine dimers form in the DNA structure that would lead to mutations? And that would have not been possible at an academic. It would be possible, but it would be a more challenging possibility at an academic institution. And so it was the opportunity to work with a collection and a diverse collection of very bright, committed, hardworking scientists that led me to stay for the last 30 years.
Okay, I guess that I will ask one last question. And I think you partially addressed this in the dialogue that we've had so far in the Q&A. You know, fundamental bioenergy research has really evolved over the past decades. What do you think the most important advances have been?
To date, it's the ability to sequence multiple genotypes at low cost. So the high throughput, low cost transformations that we've seen in DNA sequencing, coupled with proteomic capabilities, metabolomic capabilities. And then encompassing or enveloping all of that is computational sciences. And we can do things today that we were not able to do even 5 years ago as a result of advances in computational sciences. I had a picture of Dan Jacobson on one of the slides, and there was a competition to break the exascale barrier of computing. And there were, Summit was the platform at the time, just a few years ago. And the first exascale compute was actually done on Poplar data. And he won the Gordon Bell Prize for that. And so as a result of both the physical platforms and the software to drive those platforms and the large data sets that we're now generating in biology, I think the future of this, many of the things I listed as the near term are a result of those two things.
Thank you very much. Well, I think this has been another really extraordinary lecture, and I have really enjoyed this, and I think probably, thank you.
Thank you.
From tropic freeze to Chernobyl. I mean, wow. What a career.
Yeah.
Oh, what
Me
a
too.
career.
I look
Yeah.
back and think, wow,
Yeah,
so lucky, so fortunate. So thank you for the support.
thank you very much. Thank you.